development. In this we have already studied about the definition of growth, the phases of growth, the parameters of growth, and then the growth curve, growth rate, which deals with the arithmetic growth and geometric growth. Now growth is a physiological process which is permanent irreversible change accompanied with increase in the dry weight of the organism. That is the definition of growth. And there are primary growth and secondary growth. Primary growth is the increase in the length of the stem. Secondary growth is increase in the girth of the stem, that is the thickness of the stem. Then after studying the conditions for growth, that is the factors affecting growth, there are some three terms related to growth. So they are differentiation, then de-differentiation, then redifferentiation. So what is the differentiation? What is de-differentiation? What is redifferentiation? So differentiation means it is usually referred to the tissues which are present in the plant. Now whenever in the case of growth, we have studied the phases of growth, that is meristematic phase, elongation phase, maturation phase. So in the meristematic phase, the cells divide by mitosis, and these cells which are produced by mitosis, so they enlarge by the protoplasm synthesis, elongation phase. Now after the daughter cells elongate, so these newly formed daughter cells, they differentiate to form various kinds of tissues like parenchyma, collenchyma, sclerenchyma, xylem and phloem. So this is called as a differentiation. So differentiation means the meristematic tissues, when they give rise to permanent tissues, in other words. So the meristematic tissue, they divide and give rise to the cells. Those cells will differentiate. Maturation phase it is actually. So they mature to form a particular type of tissue. They perform a particular function, and that is differentiation. So this permanent tissues, when they are formed, it is differentiation. Now sometimes what happens in course of time in the plant itself, which we have come across in the secondary growth. So when we study about secondary growth, we come across that the permanent tissues, especially the living permanent tissues like parenchyma and collenchyma. Sclerenchyma is a dead tissue. So this parenchyma or collenchyma, they what happens in permanent tissues, there is no power of cell division. The main important difference between meristematic tissues and permanent tissues is meristematic tissues retain the power of cell division, whereas permanent tissues, they have lost the power of cell division. So these permanent tissues like parenchyma and collenchyma, so when they have lost the power of cell division, or they are not having the characteristics of meristematic tissue, when these permanent tissues like parenchyma and collenchyma, they acquire the meristematic properties or the characteristic features of the meristematic tissue. So then we call it as de-differentiation. So when this permanent tissue becomes meristematic, so it is called as de-differentiation. That is second one. Differentiation means the meristematic, they have become permanent. They have lost the power of cell division. Now when these tissues, they regain, they restore the meristematic properties and continue to divide, that is called as de-differentiation. So the permanent has become meristematic. Now these meristematic, when again they become permanent, that is called as re-differentiation. So already they have been differentiated here. Again the meristematic becomes differentiated, that is in the second step, that is re-differentiation. So like that, differentiation, de-differentiation and re-differentiation, these are the terms which are related to the growth. So growth actually takes place by the cell division, that is differentiation. The meristematic tissues, they divide and gives rise to the permanent tissues, differentiation, maturation. This is one. Now in the course of time, when secondary growth takes place, during secondary growth what happens, the permanent tissues, especially parenchyma and collenchyma, they undergo de-differentiation. They acquire the properties of meristematic tissue, that is called as de-differentiation. Then when that meristematic tissue again differentiate into some type of tissue, it is called as re-differentiation. Like that, during growth we come across differentiation, then re-differentiation and de-differentiation. Then related to growth, so we come across an important aspect, that is called as plant growth regulators. They are commonly called as PGRs, plant growth regulators. Plant growth regulators are also called as plant hormones, or they are also called as phytohormones. So another name for this. Now what are the plant regulators, plant growth regulators? So plant growth regulators are chemical substances which regulate growth, or in other words, they influence on the growth process. They are called as plant growth regulators. So very simple meaning of this. So plant growth regulators are chemical substances which influence on the growth process. So these, in other words, they regulate growth. They are also called as plant hormones or phytohormones. Now when we say that they influence growth, this influence or this regulation on the growth process may be beneficial or may be harmful. So that influence on the plant growth may have some benefit, or it may certainly harm, or there may be some retardation on the growth process. So like that, on the basis of whether they are benefit or whether they are not benefit, that means harmful. So that is there are two types. So this those which benefit the plant, they help on the growth of the plant. So they are called as growth promoters. So growth promoters are one which promote growth. They benefit growth. That means they have positive effect on the growth process. They are beneficial. They are useful to the plant. So that is why they are called as promoters. They promote growth. Promotion we say. Now those which harm to the growth or they stop the growth, they are called as growth inhibitors. They inhibit growth. They retard growth. So there are certain hormones, there are certain growth regulators which benefit the growth or which harm the growth process. So those which benefit, they are called as growth promoters. Those which retard growth, they are called as growth inhibitors. Now growth promoters, for example, auxins are one group of such substances, and there are also what are called as gibberellins, and there are cytokinins. So these are some examples. And inhibitors, abscisic acid is one example, and also ethylene is also another substance which influence the growth process. So growth promoters and growth inhibitors are both influencing the growth either positively or negatively. So these are on positive effect, these are of negative effect. Now this is what are growth regulators or plant growth regulators or phytohormones, or they are also called as plant hormones. So these hormones, they either promote growth or they retard growth. That is how the growth inhibitors and promoters they influence the growth. So these are some examples. Now one by one we shall study about these auxins, how they affect, which part of the plant they affect, and cytokinins, abscisic acid and ethylene. Now first group of hormones, what are called as auxins. So auxins are one group of growth promoters which influence on the growth process. They benefit the growth process. Now we shall see how, before going to the effects of auxins, so just an introductory part of the auxins, about the discovery of auxins or the general discovery of this auxins or the effect of these auxins. Now actually auxins word has been obtained from the word auxere. So auxere means to grow. It is a Greek word which as the chemicals influence on the growth process, hence they were called as auxins. Now auxins, they were first discovered. A brief introduction of these auxins. So they were first observed by Charles Darwin and his son Francis Darwin. So Charles Darwin and Francis Darwin, when they were observing the plants, especially a plant called as canary grass, so they were probably observing in relation to maybe evolution or some other structure, because Charles Darwin is mostly concerned with evolution. So when he was studying the characteristics of that plant, he was observing the plant characters. That is when he was observing the canary grass plant, when these two were observing, they noticed a chemical in the plant. They identified that a chemical which is present in the plant, or there is a substance which is produced by the plant, and that chemical substance influence on the growth process. So that is how auxins were first discovered. However, Darwin did not give the detailed nature or the detailed aspects of that substance, where it is produced and how it will influence the growth process. All that aspects he did not explain. But however, he gave a clue about the presence of such substances which influence on the growth process. So this is how auxins were first discovered. But later experiments were conducted about the presence of this substance in the plant. So two scientists went and went. FA went and FW went actually. So they observed that this chemical substance is present in the tips of the stem and also tips of the root. So at the tips of the stem they are present, and they confirmed the presence of this. And later another scientists, Boyson and Johnson, they conducted experiments on a plant which is called as avena, which is commonly called as oat. So the experiments which were conducted on this, they were popularly known as avena curvature test. That plant which they selected was avena plant, and in this they conducted certain experiments in related to the auxins by the confirmation of the auxins, and those experiments were came to be known as avena curvature test. So this what they did is they took the coleoptiles of these avena plants, that is the newly germinated seedlings of this plant were selected. So this newly germinated seedlings which had coleoptiles, and this is the coleorhiza. So this coleoptile, this is the coleoptile. So the coleoptiles were selected for this experiment, and when these young seedlings were placed in dark and provided with light, unilateral light, means from one direction, there was bending of the stem towards the light. By this they confirmed that the bending of the stem towards the light is because of some substances present and produced at the stem tips. That is what actually Boyson and Johnson identified. Then later what they did is they removed the coleoptile tips. So the coleoptile tips was removed, and when light was provided from one direction, there was no bending of the stem towards the light. So the bending of the stem was completely absent, and this is because the substances which are produced at the tip of the stem, as the tips were removed, so hence that bending was not influenced by the light which was provided from one direction. And later what they did is they placed an agar block on this cut end and then placed the coleoptile tips. The coleoptile tip was removed, but the tip was placed on the cut end of that coleoptile with a small piece of agar block. So a small agar block was placed there, and on that the coleoptile tip was placed. Now the coleoptile tip which was present here, so now there was bending of the stem towards the light. So when the agar block was there, when the coleoptile tip was there, there was bending. But when the coleoptile tip was not there, only the cut end of the coleoptile did not show bending towards the light direction. This shows that a substance which is, or in other words, the bending of the coleoptile is caused because of the substance produced at the coleoptile tips. So even if the agar block is placed, the substance which is produced at the coleoptile tip, so that substance diffused through the agar and caused influence on the lower part of the stem, and thus the bending of the plant or the bending of the stem was seen in the agar or of this stem. So this is a Avena curvature test which was explained by Boyson and Johnson. So this shows that these auxins, but even then the chemical substance was not named as auxin. So later it was these substance was named as auxin. So auxins were named so because auxere means to grow. And even another scientist called as Kogel, he also observed that this chemical substance is also present in the excretory product of human beings. So there he identified about 40 milligrams of such substance was found in some 100 ml of such excretory material. So that is where even auxins are present in such source also. That was first identified this by Kogel. So later these substances were named as auxins. And auxins, when they were studied, and the chemical nature of the auxins were found to be identified, and they were found to be indole 3-acetic acid. So this indole 3-acetic acid is the chemical name of this auxin. So indole rings. So to the third position of the indole ring, acetic acid is attached, and that forms indole acetic acid. And even there is one more type of auxin which is called as indole butyric acid, also called as IBA. So these are the two natural auxins which are found in the plants. The most common is IAA, indole acetic acid. Indole butyric acid is also found in plants, and this is a natural auxins which are found in the plants. Similar to these natural auxins, there are certain synthetic auxins which are also produced artificially. Now see, these synthetic auxins, they are used for various agricultural and horticultural purposes. Now for example, naphthalene acetic acid is one artificial auxin which is called as NAA. This is one naphthalene acetic acid. And 2,4-dichlorophenoxyacetic acid. So this is another which is used commonly for various aspects in horticulture and agricultural purposes. So naphthalene acetic acid, NAA, 2,4-dichlorophenoxyacetic acid, and even there is one more 2,4,5-trichlorophenoxyacetic acid. It is called as 2,4,5-T, it is referred as. And in addition to this, there is indole propionic acid, indole pyruvic acid, but these are not called as IPA because there are two types here, indole propionic acid, indole pyruvic acid. So these are also artificial auxins. So like this, there are still many more types of artificial auxins which are practically used in agricultural and horticultural purposes or the gardens. Now these synthetic auxins, they are used for various purposes or applications which we can say. So there are what are called as the practical applications of auxins. So for what purposes these auxins are used? So practical applications are also called as the physiological effects of auxins. So what are the practical applications or what are the physiological effects is one and the same. Now especially the synthetic auxins are used for these purposes. Now natural auxins are present in the plant, but the extraction of these auxins is somewhat found to be costlier and again used for the various purposes. So we can also use natural auxins for other purposes. But the removal or the extraction of these auxins or these chemicals from the plants requires a certain steps there, certain process, and that is why as a substitute these natural auxins, these synthetic auxins, artificially prepared auxins are there, and they are used for various purposes in the agriculture. So one thing is, in the root initiation, these practical applications are used, and also it is used in the apical dominance, and it is used in the what is called as parthenocarpy. So like that, prevention of abscission layer. These are some uses of auxins which are used for various purposes, and also they induce flowering in certain plants. So root initiation, one by one, what is this, how they are used, just in brief, how the auxins like NAA, then this other auxins are used. Then even this is used in as a weedicide. Now in the most cases, NAA is commonly used for the practical applications. Now root initiation, to initiate roots, to produce roots, this is the meaning, to induce roots in a plant. Now naturally roots are produced by themselves. There is no question of producing roots artificially in that. So when a seed is placed in a soil, naturally it will germinate and produce its own roots. So that is natural. But artificially we can produce roots in stem cuttings, especially. Just by cutting a mature stem and place in the soil, it will not induce roots. But it is true for some plants. For example, in case of hibiscus, in case of rose, or in case of jasmine and all that. So in these plants, we can place or we can induce roots just by taking a stem cutting of hibiscus or stem cutting of rose and place in the soil and provide proper water and all that. So the stem automatically will produce roots and grow into an independent plant by initiation of buds, branches and all that. That is a natural root initiation. But in other plants which we want to produce roots, we can make that stem to produce roots by the help of these auxins. Now take for example a mango or a tamarind or any tree plant or like that. So if you cut a mango stem or a neem, Bevingida, or a tamarind and place in the soil, it will not produce roots. So if we want to grow a new plant, we have to grow it by the help of seed, isn't it? But instead of that, by making the stem to produce roots, we can make it with the help of auxins. Just we have to take a solution with the known auxin concentration. So a solution containing of a particular concentration of auxin, especially NAA. Now this auxin concentration which is taken, in this the stem cuttings are placed. Suppose if you take this in a beaker and place the stem cuttings in this for some hours, for some period, these will absorb the auxin content from the solution, and then what happens, they start producing roots from this ends. So when these stem cuttings they start producing roots, so we can take that stem cutting and place in the soil, and later what happens, these stem cuttings will produce buds and they will produce the branches. So that means we can make the stem cutting to initiate roots and thus we can grow them into a new plant. This is the use of auxins. And then apical dominance. Apical means top, apex. Dominant means which is having more influence. So this apical dominant means what? For example, if you take a common plant, any plant like this, suppose this is the plant, this is the apical bud and here there are the axillary buds, lateral buds. So this is the apical bud. Now naturally you might have experienced whenever you remove the apical bud in a plant. So imagine that a plant is growing straight, almost there are no branches. So when it is growing in its height, you remove the apical bud of this plant. So when you remove the apical bud of this plant, automatically the lateral buds will sprout into branches. So this is because the presence of apical bud will not allow the growth of the lateral buds. In other words, the apical bud will inhibit the growth of the lateral buds. So this is all because of the auxins. So auxins which are present at the tip of the stem, they diffuse in the plant and they suppress the growth of the lateral buds. They do not allow the growth of the lateral buds into branches. So when you remove the apical bud, you are not supplying, or the supply of the auxin is broken down. It is cut off, in other words. So the supply of the auxin is cut off to the lateral buds. So there is no supply of the auxins to the lateral buds, in other words. So when there is no supply to this of the auxins, so these lateral buds will grow into branches. This is what is called as apical dominance. That means the apical bud is dominant over the lateral buds. So this apical bud, when it is present, it will not allow the growth of the lateral buds. That is the meaning. So apical bud is always dominant over the lateral buds, and when apical bud is removed, the lateral buds will grow into lateral branches. Now this is the meaning of apical dominance. Now how this is applied? This by the use of auxins. This phenomenon is applied for the storage of potatoes and also even onions. Especially potatoes are used for this, and also onion also is used. Onion tubers, onion bulbs are used for the storage. Now in the potatoes, take for example a potato. In the potato there are small depressions which are actually referred as eyes. So these potato which are having small depressions called as eyes, when you place a potato in a cool climate or in a moist place, you can see the potato becomes somewhat green and small bud-like structures are produced from these regions. So what is this? Actually potato is a stem modification, remember. So in the stem modifications we study this. So potato tubers, what we use, they are the modifications of stem. So these regions which we call it commonly as eyes, they are actually the regions of lateral buds or the axillary buds. So because so when you place the potato in a cool moist place, we can see small bud-like structure appearing on this region. Now this gradually becomes large and grow into leaves like this. So this is actually the lateral bud which grows there. So that means the auxins which here what we have studied here, the auxins when supplied to lateral buds, they stop growing. When there is no auxin, the buds will grow. So here these potatoes, when they produce the buds like this, it is because due to the absence of the auxins. When there are no auxins, the lateral buds grow. Here also when there are no auxins, the lateral buds grow. So if the lateral buds grow on the potato, so this potato is actually somewhat not properly, means it cannot be consumed properly, or it should not be consumed, in other words. So that means the buds which are produced here, when they produce, we can grow them into a plant. If you remove this part, they will grow into a plant there. So to suppress the growth of the buds, if the buds grow on the potato, the value of the potato will be reduced. It will not have value, nutritional value will be reduced, in other words. So that is why we should not allow the growth of these buds on the potato. So these buds can be suppressed by the addition of, or by the spray of auxins to this potato. So potato tubers can be stored for a long period by spraying auxins on the potato, and that too known concentration of auxins. So when auxins are used in excess, it will show adverse effect. So the one point to just remember here, when I was telling about root initiation, I said that when we take auxin solution here and place the stem cuttings, roots are formed. This solution should be of a known concentration. So we cannot take any amount of auxins in that solution. There is a particular measurement for that, which we are not supposed to go into that detail. So here we have to take a particular concentration for a particular amount of water. So for 100 ml of water, there is some particular concentration which we have to take. Suppose if you take excess of auxins in this, it will not produce roots. So excess of auxins also will inhibit the root initiation. See, under known concentration there will be root initiation, but under excess of auxins there will be no root initiation. Instead, excess of auxins will inhibit the formation of the roots. So like that here, the auxins which are sprayed to the potato tubers for the storage, it is also of known concentration. Excess of auxins should not be sprayed to the tubers. Like that, potato tubers can be stored by spraying auxins to the potatoes. When we spray auxins, these buds will not develop, and hence these potatoes will be kept as fresh as they are, and they can be consumed without any doubt there. That is the use of these auxins. So today also potatoes and onion bulbs and such kind of tubers, like even beetroot and all those are stored in large scale by spraying auxins. Now we cannot store potatoes in our house in a large scale. Suppose assume that if potatoes are very cheap in the market or onions are very cheap, we cannot buy a large quantity and store them in our house. So if you do so, most of the potatoes will germinate, will produce these buds, and these potato tubers are spoilt. They cannot be consumed. They are of less nutritive value, and that is why these potatoes are sprayed with the NAA. So even we also cannot spray NAA because these auxins, which are synthetic auxins, they are somewhat costly, and only when we do this in a large scale, so it will work out. That means it will become applicable because in our house we store only little amount of these quantity. So this is what is called as apical dominance. So that means we can this phenomenon is applied for the storage of potatoes and onion bulbs. Then parthenocarpy. Parthenocarpy means it is the production of seedless fruits. Production of seedless fruits is called as parthenocarpy. Now, what is the advantage of seedless fruits? That is question here. Fruits with the seeds and fruit without seeds, difference. So in case of grapes, we know that there are seeded grapes, there are seedless grapes. Seeded grapes, they are somewhat not comfortable for eating. So when we eat seeded grapes, we have to remove the seeds in our mouth and then we have to throw them. But seedless grapes, we can easily, without any difficulty, easily we can swallow them or easily we can consume them without any difficulty or such kind of problems we don't have for swallowing. So that means the seedless fruits are more comfortable or more beneficial than the seeded fruits. And these seedless fruits, they are only applicable for small seeds. But for big seeds, it is having advantage. But small seeds, even if they are seeded, there is no problem. For example, take tomato. Tomato is having seeds. So today we are eating, generally we eat tomato along with seeds. That means seeds do not become a problem for us for eating. So if the same seeds are large, they will become somewhat uncomfortable for our eating. So like that, seeds when they are large, such kind of fruits should be seedless. Seeds when they are small, for example, tomato, brinjal, then even guava plant. So all the fruits, they, even if they are having seeds, there is no any problem for eating. We can comfortably eat them. But large seeds are somewhat uncomfortable. Suppose if take watermelon or sapota or like that, if that fruit is seedless, it is advantage for us. So here flowers of some plants, this is not applicable for all plants. Auxins when sprayed to some plants, they produce seedless fruits. So these seedless fruits are by the influence of spraying auxins to the flowers which produce fruits without seeds. That phenomenon is called as parthenocarpy. So parthenocarpy is the production of seedless fruits by the spraying the auxins, again of known concentration. So always whenever we use auxins for any application, they are of particular concentration should be used. So when there is variation in the concentration, we will not get proper result. So that result will be somewhat having adverse effect on the that part. That is parthenocarpy. Then prevention of abscission layer. Now what is this abscission layer? Now suppose if you take a leaf or if you take a fruit. So in some plants we might have observed. So almost you might have observed when fruits are small, we see these fruits, they fall down due to some reasons. In coconut plant also you might have seen small coconut fruits, they fall from the plant. And even in case of mango or even in papaya plant, you might have seen. So in mango plant, when you have seen, when the mango fruits are very small, we can see some of the mango, these small immature fruits, they fall down, and that means it is somewhat a disadvantage to the plant. So and also it is a disadvantage for the consumer there, what we use. Now why these fall? Again question here. Why these fruits when they are small, when the coconut is of certain size, when the mango is of small size, why they fall? It is because suppose this is a fruit, this is the stalk of the fruit. Here there is a layer which is formed in the stalk of the fruit. A dead cell layer is formed there. So where this stalk becomes weak and this fruit will break at that point. So when this becomes weak, it will break at that region and the fruit will fall at a immature stage, premature stage we can say. So this is called as abscission layer. Abscission layer is a layer of dead cells where the stalk of the fruit becomes weak and it will break at that point. So the fruit will fall at premature stage. So this fruit when it falls, it is useless. So when a coconut is of small size, if it falls, no use. If the same coconut remains in the plant, it becomes large, one or the other day, it will be useful by some person. So we can use that fruit. Even a mango which is of small size, if it falls down, it will be no use. If the same mango remains in that plant, it will grow large and it will be useful. We can use that. We can consume that, isn't it? So that is why this abscission layer can be prevented by spraying auxins again. So if the fruit falling, if the premature fruit fall takes place, if leaves fall, it will not affect us. So that means we are not using the leaves. Suppose if the leaves fall, we think that again new leaves will grow and the plant will get more leaves. But fruits, they will be, once they are formed, again we have to wait for the next flowering season, isn't it? So that is why especially the formation of abscission layer in the fruits, premature fruits, it will result in the premature fall, and thus we can prevent the formation of this abscission layer by spraying known concentration of auxins to these plants. And again we can also induce flowering in the plants. So flowering can be induced by the spraying of auxins. Then also this flowering can be done only in certain plants, not all, and that is induce the flowering or production of flowers. Then one more use of this before going to this. In case of fencing, this apical dominance is used especially. So when certain plants are grown in the fence region or at the border, we can grow them, made them to grow many branches by this apical dominance. We can go on removing the apical bud and make the plant to grow, produce more branches, so that the plant will grow into a bush like, or it will spread and it will almost block that area and it will become a proper fencing to that region. That is also in case of fencing, or which are also called as the hedge plants. They are called as plants which are grown in the fence. They are called as the hedge plants. That is the hedge plants there. And lastly, the plants which are used as weedicide, especially 2,4-D is used as a weedicide for this. See, almost in all applications, NAE is very common, naphthalene acetic acid, but particularly 2,4-D and 2,4,5-T is used as a weedicide. Now out of these two, this is most commonly used, 2,4-D. Now 2,4-D is a weedicide, one which kills weed. Side means to kill. So whenever we have the word ending with side, it means to kill. Suicide we call as, means a self-killing. When a person kills himself, we refer it as suicide. So here sui means actually self, killing himself or herself is called as suicide. So like that, insecticide, a chemical which is used to kill insects, pesticide, bactericide, fungicides, they are all agents which are used to kill a particular group of organisms. Like that, one which is used to kill a weed. Weed, what is weed? First we have to see. We have to know what is weed. A unwanted plant in a crop field is called as weed. So in a crop agricultural field there, so when we remove certain plants which we are in not need, if you are growing groundnut, if you are growing jawar in that field, we usually remove certain plants which are not necessary other than that crop. So it may be grass or any other plant, need not only compulsorily grass, any plant may be a weed. So these unwanted plants in the agricultural crop fields are called as weeds. So to kill these weeds, auxins are used. So just by spraying 2,4-D in a field, especially in case of monocot plants where monocots are grown like jawar, wheat, like that, not cotton, dicot weeds can be killed. See, weeds are also specific killing. They do not damage the crops, they do not harm the crops, they only damage or they only destroy a particular group of plants which we call them as weeds. And so 2,4-D is commonly used in monocot plants where they are grown. So dicot weeds are easily destroyed, and thus weedicide 2,4-D is the commonly used weedicide than 2,4,5-T, because this is more poisonous than 2,4-D. So the reason is, suppose if 2,4-D is sprayed to the field, so even if cattle feeds on the grass or any other weeds there, so it will the harm or the damage is not considerable there. It will not be more, but it will be damaging to the plants there. But 2,4,5-T is highly toxic. So that is why mostly or practically this 2,4,5-T is not sprayed to the fields, and 2,4-D is commonly used in the fields. So these are the practical applications of auxins or physiological effects of auxins, which is usually asked for 5 marks questions. So explain the practical applications of auxins or explain the physiological effects of auxins. So any of these 5 if you mention with the 1 sentence each, it is more than sufficient. So name the natural auxins, short questions, name the synthetic auxins, these are also asked. So the synthetic auxins, naphthalenic acid, 2,4-D are the most commonly synthetic auxins. So these are the practical applications of auxins. So in the next part we shall study about the gibberellins.